All right, welcome to WSQ number 6. This one will be combining two sections, Chapter 2, Section 4, and Section 5, on basically human vision and optical tools. And so we're going to kind of learn a little bit about how the eye works, which will definitely give us a little precursor into the future when we talk about human anatomy in the spring. And we're also going to talk about the different optical tools that science uses in understanding and studying how light actually is used in vision. And all this stuff's really important because this is going to help us as we transition next chapter or next section into dealing with microscopes and understanding how we see things up close in the world of cells. So let's get started. We're going to learn today how do you see objects, and we're going to learn what type of lenses are used to correct vision problems, which those are two pretty big ones. And then ultimately, how are lenses used in cameras and telescopes and microscopes and mirrors as well in those optical tools that we use. So let's start with a quick overview of the eye and vision. Your eyes respond to a stimulus that we know as light. And what the eye does is it actually converts that stimulus, that light, into impulses that travel along your optic nerve that your brain interprets and enables you to see. Let me just say this. Human sight is a miracle. It's an incredible, incredible thing. Well, it's truly not a miracle because it's natural and God's designed it in that way. But to think that human sight could have come through random chance over billions and billions of years, millions of years, and random genetic, deficiencies in heredity that would lead ultimately to this is just incredibly absurd to me. When we understand the intricacy of the human eye, it's incredible. So if you look at this eyeball here, we see some really important terms: the cornea, the iris, the pupil, the lens, the retina, the optic nerve. And we're going to kind of talk real quickly about what those different parts of the eye are. The cornea, that first section we looked at, that's the transparent front surface of your eye, and that's used to protect the eye. And it's also the first of two lenses that actually focuses light. If you look at it, the cornea has got a rounded shape to it, which gives it a perfect ability to pinpoint and focus light into one single portion. The pupil is the opening of the eye, and it's just a circle basically of the eye that lets light in. It's hollow, it's open, and the pupil actually has some muscles that are attached to it through the iris that allow the pupil to expand or shrink depending upon the amount of light. So if you've ever woken up in the morning and your mom pulls back your blinds real quickly, what would happen if we could look at your pupils pupils would actually shrink. They would contract quickly as they try to keep all that light from getting in the eye. That's also why if you've ever been to the eye doctor, and they dilate your pupils stay big. And what they're trying to do is be able to actually look at the back of your eye and test for health reasons. The iris is just a ring of muscles that change the pupil size, and God could have just made the iris bland. The iris could have been just bland and all one color, but instead he made it a variety of colors, and so we get pupils from green and brown to blue. The lens is behind the pupil, that opening of the eye, and it bends light to form an upside down image that actually strikes the retina. And the retina is these cells known as rods and cones that are on the inside back of the eyeball, and these little receptors actually transmit that signal, that light, that image to the optic nerve, which sends it to the brain. The rods are sensitive cells that respond to small amounts of light. They actually are dealing with non-color, so black and white, so they're very, very sensitive. Cones are cells that respond to color, red, blue, and green. And so if you have damaged cones, or if your cones don't work, you can be colorblind. If you have rods that are not functioning correctly, you can actually be blind completely, which happens. There's a really cool dissection of the eye. If you go to phschool.com and go to CGP 4153, it's a really cool activity for you to check out. It's a virtual dissection of the eye that actually shows those different parts. Now let's talk about some of the diseases that can affect the eye, or not necessarily diseases, let's say the conditions. There's two types. If you wear glasses, you're either nearsighted or farsighted. And what happens, it's really interesting. In nearsightedness, the eyeball is just too long. And the problem is, because it's too long, we'll notice the focal point actually happens short of the retina. So instead of the light converging the focus point on the back of the eye at the retina, it actually converges in the middle of the eyeball, which means that you're going to get a blurred image. Okay, they're nearsighted. So in a farsighted, notice the eyeball, instead of being too elongated, it's actually too short. And so what happens is the focal point never happens. It converges behind the retina when it should be converging on the retina. And so both of these are treatable. They can use glasses. We can actually correct them. Okay, well, let's show you how we can correct one. In nearsighted, how do they correct? Well, the problem, of course, is that the focal point is in front of the retina. Okay, so what they'll do is they'll actually use a concave lens that's used at the front of the eyeball to correct nearsightedness. You'd use this with either they could use a corrective lens like I can't remember what they're called now. People that wear glasses, or they can also wear contact lenses. There we go. Which actually allows them to go ahead for a nearsighted. Eyeball's too long, so by using a concave lens, it shortens the eyeball and actually protects or corrects the eye. If it's farsighted, okay, so here we go. Here's a nearsighted example. They use a concave lens. Notice what happens is it causes the light to reflect back a little bit wider, and then when that light comes in through the lens, it actually lands correctly on the retina. In farsightedness, because the eyeball is too short, they'll use a convex lens that actually starts the process of that light converging before the lens, and you get it landing perfectly on the eyeball. That's why when you go into the eye doctor, they're going to have you do some tests, and they're going to give you a prescription, and that is your prescription for your eye, because if they use a different prescription from what's correct for yours, then it wouldn't do any good. It wouldn't work for you, and so it wouldn't solve the problem. So they use concave lenses to correct nearsightedness and convex lenses to correct farsightedness. Now, how else have scientists used lenses besides just correcting vision? Well, they've also used them in different optical instruments. I want to talk about three major optical instruments. The first one is the camera. If you've ever seen a camera, what a camera basically does is it mimics the human eye. The camera focuses light through a lens, okay, right here. It's called the object lens. That light goes through a shutter, right, okay, and actually projects onto either film, in the case of an old camera, 35 millimeter, etc., or in the case of digital cameras, it actually is on a little sensor that kind of mimics what happens in the eyeball. And so what happens, rays bend the light as they pass through the lens, so that focuses them back so that they will actually land on this film. And what happens when the shutter is released, it opens this just for a second so that the light can reflect and it can be expose the film on the old 35 millimeter cameras, and that's how you get that image. Okay, that's actually exposed on the film. In a digital one, it's exposed onto that particular card. Now, in the case of our second object, we have telescopes. A telescope's job, a camera, is to record an image. Telescopes actually form enlarged images of objects that are much further away than we can see with the human eye. So they'll use lenses or mirrors to collect and focus light from distant objects. In a refracting telescope like this one, what it's actually using is a pair of lenses. So it has a convex lens at the barrel of the telescope, and then it's got a convex lens also down here at the eyepiece. And what happens is as it gathers light, it can focus it in. We can see distant lenses or distant objects like the stars much closer and much more accurately than if we use the human eye. You can also use a reflecting telescope. Reflecting telescopes, instead of refracting the light or using lenses, they'll do as they say, they use mirrors. So they'll have a concave mirror, that's a big concave mirror at the back, an opening at the front. The light hits the concave mirror and then gets reflected off of a plane mirror and through a convex lens, which allows us to see much closer objects in the distance. The third type is the one that's most important to us, and that is going to be microscopes. A microscope is an object that uses a combination of lenses to produce and magnify an image. This object to the right, this example of a light microscope, it's actually using just normal light. The ones we're using have produced light. So instead of having a plain mirror on the bottom, the microscopes we use will have an actual light source like a little light bulb. But you'll notice there will be an eyepiece, which is a convex lens, and then there will be objective lenses. There's usually multiples. Our microscopes have 3 different lenses, and we'll be going over those together. They've got a stage in which you can put your specimen, and then it allows light to travel through the specimen, which allows us to look at small objects close up. So microscopes use a combination of lenses to produce and magnify an image. And the magnification is just the ability of microscopes to make things look larger than they actually are. Okay, and magnification is great because it allows us to study in more detail objects that normally would be too small for us to study. Resolution on a microscope is the ability to clearly distinguish the different parts of an object. So you can have great magnification, but if the resolution's not high, then it's going to be hard to see and decipher the distinct differences between parts of an object. And one thing to understand too is there are different microscopes. There's the light microscope and the electron microscope. Electron microscopes, actually, light microscopes are just passing light rays through it, and those light rays carry that image to our eyes. In the example of an electron microscope is they actually use tiny particles, electrons, that basically take pictures of extremely small objects. We're going to have an electron microscope coming in probably this later on, and we'll get a chance to see what that looks like. It's pretty awesome and pretty close up. So light uses light to expose, and electrons use electrons to produce a magnified image. All right, that is it for WSQ number 6. I hope you enjoyed it, and summarize, write your questions down, answer those on Haiku, and I will see you guys